everyone. Thank you so much for joining us for the brown bag today. We have two speakers. The first will be Brendan Talwar, followed by Taylor Armstrong. To give you a little background about each one, Brendan graduated in 2011 from Furman University. After that, he jumped into doing research and some teaching as well before going to grad school for ecology and evolution at Florida State University. To tell you a little bit more about Taylor, she graduated from the University of South Carolina with a bachelor's in biology in 2013, and then went on to VIMS for graduate school to study marine science, graduating this year. A fun fact about both of them is that they're both from Louisville, Kentucky, and Taylor's dad actually taught Brendan math in school, which is pretty cool that they both signed up to present today. Brendan will be kicking us off, talking about discard mortality of deep sea sharks. And just to follow up, to let you know, Brendan is currently a fellow with the Marine Mammal Commission, and Taylor is a fellow in the Ocean Acidification Program this year. So let's give them a warm round of applause.
Thank you all for coming. I guess any stats problems you can direct towards Mr. Armstrong. It's not my fault for those. But I just want to say it's really great to be here. When we first conceived of this work, our audience was really made up of some of the folks in this room more than anyone else, so it's really nice to actually present here at NOAA for research that we hope is actually very applicable. So yeah, I'll be talking about discard mortality of deep sea sharks. This was with folks at the Cape Luther Institute as well as the New England Aquarium, and then my advisor dean at the Florida State University Coastal Marine Lab. And so I just want to recognize them before we get started. They've been really great mentors over the past few years. So I'm going to frame this research in the context of bycatch, which is truly a global problem. It's estimated that about 40% of everything caught in global marine fisheries can be considered bycatch, which by this definition means that it goes unused or unmanaged. So typically a lot of these organisms are thrown back, oftentimes alive, and what happens to them afterwards is generally unknown. So to bring that close to home, this is an example from shrimp trawlers in the Gulf of Mexico. It's a little outdated. I think the bycatch is a bit less than it is in this figure. But back at this time, for every 1 pound of shrimp caught, up to 10 pounds of marine life were thrown away. I think now it's around 5 to 7 pounds, which is a little more accurate. But you know, bycatch is a problem not only for fisheries managers trying to sustainably manage our deep sea resources, but also fishermen. It represents a huge cost to fishermen, a big waste of time and resources as well. And so it's something I think we can all agree is better if we eliminate it entirely. Unfortunately, this issue of bycatch is becoming more and more relevant for fishes that are caught in the deep ocean. So you can see here from 1950 to the mid-2000s, the mean depth of fishing for commercial bottom fishing globally has gone from about 200 meters to over 500 meters deep. And this is from Watson and Murata 2013. And that's primarily due to two reasons. One is overexploitation of shallow water stocks. Again, this isn't just U.S. fisheries. This is directed, more of a global context. And then improved technology as well. It's become really, really easy to target deep sea fish when previously we couldn't even locate them. So deep sea organisms are generally characterized by a few things that make them ill-suited to exploitation. At the individual level, they have low metabolic capacities as well as low energy stores. So if you think about that in terms of a capture event, it means that they'll get exhausted very quickly when they're caught on a long line or in a trawl. And at the population level, they're case selected. So things like this blunt-nosed sixgill or a big-eye sixgill are characterized by long gestation periods, long lifespans. They generally have few offspring, and they have a late age of maturity. So again, characterized generally not for all deep sea organisms, but for many that have conservative life histories that make them ill-suited to exploitation. Deep sea chondrichthyans, meaning sharks, skates, rays, and chimeras, are kind of at the extreme end of this spectrum. So some families like squalids and centrophorids, your dogfishes basically, they have population doubling times of over 30 years. We still don't know much about most of these species. We're finding new deep sea sharks all the time. We're finding new shark species all the time. Put that in perspective, I think since 2000, about 65% of the 200-something species that we've discovered are actually considered deep sea taxa, which means they spend all of their life below 200 meters deep. And actually, most people don't know, but actually about half of all chondrichthyans, period, are deep sea, meaning they spend their life below 200 meters deep. So they're very data deficient in general. A lot of taxonomic uncertainty in this group. Oftentimes when we catch a deep sea shark in the Gulf of Mexico or even the Bahamas, we don't know what species it is until we get some kind of genetic confirmation. And a lot of those times it's something that we've never described. So to kind of wrap up that portion, basically we don't know a whole lot about a lot of these species. We do know that they're ill-suited to exploitation, and unfortunately fishing pressure has progressed into the habitats where you might encounter them. So gulper sharks are kind of the best example of everything I've talked about so far. Again, this genus Centrophorus. They have among the lowest reproductive potentials documented to date. So for some of the species, this is a big species group, but for some of them, they might only mature at 15 to 17, maybe 20 years old, only have 1 or 2 pups every 2 years. They can live 50 to 70 years. So as you can imagine, when they're exposed to fishing pressure, they might experience significant declines in catch rates or population size over time. And there are some data to back that up. This is from Graham et al. In 2001. You can see there's mean catch rate on the y-axis, and then there are these 3 different capture locations off the coast of Australia: Sydney, Ulladulla, and Eden. And you can see catch rates, mean catch rates here from surveys taken in the late 70s. This is from ships that were trawling. And you can see that catch rates were highest in this 3rd depth zone, around maybe 500 meters deep in 76 and 77. Then they redid these surveys in the late 90s, and catch rates actually declined by over 95% for both Centrophorus harrisoni and Centrophorus uveiato. So it's pretty interesting because these species, for most of that 20-year period, were subjected to pretty regular trawl fishing efforts. In the early part of those 2 decades, they were mostly discarded because they had low economic value. But actually, over time, they did develop a market for liver oil, which is quite valuable, and you can get from these deep sea sharks readily. So it begs the question: if a lot of these animals, in this situation, were being thrown back alive for much of this time period, and yet you see these huge declines in catch rates, are they actually surviving after they're potentially thrown back alive? So what we know about capture-related mortality generally comes from the shallow water environment. We know that at vessel, and if it says PRM refers to post-release mortality, is species specific and it's highly variable. So I like to use these 4 shark species as an example because you can catch all 4 of them on the same long line. So we've got a tiger, a bulla, a blacktip, and then various hammerheads. You can catch them on the same line in the same habitat, same day. So basically you hold a lot of these capture-related characteristics constant, and yet they experience very, very different capture-related mortality rates. So tiger sharks might be 0 to 5%, bull sharks closer to 20, blacktips 30%-ish. This is, kind of generally speaking. And various hammerhead species are very vulnerable to capture stress. In my experience, capture-related mortality of 40% and up in some cases. Now that's a very simple example. When you actually incorporate all of these variables that play into a capture event, it becomes a very murky picture. So that's just referring to fish size and species, right? But if you imagine, this is from Davis 2002, if you imagine the path a fish takes from the time it's captured, brought up to the boat, and then released, there are a lot of factors actually playing into what's happening to that animal. So you have things like the capture event, different kinds of hooks, for instance, different length gangins, different types of line setups, basically, seawater temperature and light conditions. For animals caught in the deep ocean, it'll be very dark, it'll be very cold. That affects a whole suite of characteristics that are kind of inherent to the species being captured there. Then they're pulled up to the surface. All of a sudden, they're in a lot of light. It's a lot warmer as they go through the thermocline. Might be exposed to different sea conditions that lead to physical injury. Might be exposed to air, various handling practices. For a lot of deep sea sharks, they actually have dorsal spines on the first and second dorsal fin. And so many fishermen just don't want to risk getting stabbed by them, basically. And so instead of taking the hook out of the mouth, they'll actually rip the hook out and break the animal's jaw. So even if it's thrown back alive, it might have all these injuries. It's obviously been exposed to a lot of stress. And then it has to not only get released from the boat, but also make it back to its normal depth range. And to do that, it has to go through a lot of open space that makes them very vulnerable to predation. So based on this, you can basically think of, the way we thought about it was the deeper the animal's caught, the more stress you can imagine will accumulate over time, and that would lead to higher capture-related mortality rates for deep-sea species as opposed to shallow water species. So why does this matter in a management context? Basically, if you're trying to estimate the total mortality for a given population of sharks, it's easy to account for the ones that are landed and the ones that you bring on board. It's It's easy to account for the ones that are discarded dead, and it's easy to record the number that are discarded alive. But actually incorporating what happens to the animals afterwards can change your total mortality estimate quite dramatically. So if you assume, for instance, that all these sharks are going to live and many of them die, then you're underestimating total mortality. So it's important to incorporate these percentages into that larger equation. So our current management for elasmobranchs, and again, this isn't directed at U.S. fisheries. This is really speaking in generalities kind of across the world. Typically, at least in the U.S., deep-sea sharks aren't really targeted that heavily. There are some that are caught in the Gulf of Mexico, but not a whole lot elsewhere. So most of these fisheries are taking place outside of the U.S. But again, if you look at fishing mortality, number dead at vessel, number dead post-release, for deep longline fisheries, you might assume that most sharks are going to survive because longline capture is much less stressful than capture in a trawl. If you're discarded alive from a trawl, you might assume 100% post-release mortality. The animals have been compacted for a long time and haven't been swimming. Condition will be lower. But we know that these are probably not a reflection of reality because of this interspecific variation and all these differences in fishing practices that will result in widely disparate at-vessel and post-release mortality rates. And even just looking at the body plans, the morphology of these various deepwater shark species, you can predict that their response to capture is going to be a lot different. So that brings us to the objectives of our study. We wanted to first estimate a post-release mortality rate, and then develop a method to predict that mortality, and lastly, investigate post-release behavior. So here's the coast of Florida. We were operating on the island of Eleuthera in the Bahamas. Zoom in a bit, we were fishing in 500 to 1,000 meters right off the coast. Our two species of interest, the first was the Gulper shark in the genus Centrophorus. It's among the most highly exploited groups of deep-sea sharks worldwide. Again, that's for their livers because of the liver oil, which actually can be used in high-grade machining, cosmetics, and pharmaceuticals. They're also caught as bycatch in a number of fisheries. And then the Cuban dogfish, Squalus cubensis. This is actually the most encountered squalid in two fisheries in the northern Gulf of Mexico. So the tilefish fishery and deep reef fish fishery, where over 95% are discarded alive. On top of that, they were the most abundant species at their given depth ranges at our study location. So what we did was we set long lines at 450 to 900 meters deep. We hauled those lines after 2 to 4 hours, took various measurements, took a blood sample, ran that blood through a variety of instruments to look at basically quantifying stress of the animal. We also took a fin clip from a unique location. So let's say we put 3 sharks in a cage. We take a fin clip from the first dorsal on one, second dorsal on another, and tail on another, so we could identify those on our videos. We actually had a cage hanging at the back of the boat. We put the shark in there right after taking measurements in blood, and then we dropped the cage to the bottom, again, as close to capture depth as possible, maybe 500 to 1,000 meters, depending on the species. And each cage had 2 lights and a camera that were synced to record for 4 minutes every half hour for 24 hours. And so it gives us these little snapshots of in-cage behavior of these animals over time. So I'm going to show you a little video if it works here. So we set these long lines again, usually around 500 to 800 meters. You're catching the dogfish a bit shallower than the Gulper sharks. Again, we haul after 2 to 4 hours. All of this was, of course, taken on the nicest days we were out there. This was all done with island school students, so our high school students that would work with us for the entire semester. It's pretty low tech. We coiled everything into buckets, so it was a lot of fun. A lot of folks vomited into the buckets. And then we take these, you can see the Cuban dogfish there is quite small. Sorry, it's not really playing smoothly. So that's our cage at the back of the boat. We toss it in. We give it a release condition score or vitality score I'll talk about later. And then we drop those animals down to the bottom and get these little snapshots over time. After 24 hours, the cage is hauled to assess survivorship and get that video footage back. And also note that there was a little dissolving link on a door to the cage. So after about 22 to 23 hours, the door could fall open at the bottom, and any surviving animals could swim out. Anything that did die, we pulled back up, and we took samples that are being used in other studies elsewhere. So we caught a bunch of different species. I'm just going to focus on these 2. You can see we caught a lot more Cuban dogfish than Gulper sharks. They're also a bit smaller, and we caught them at shallower depths on average, where it was also a bit warmer. So I'll talk a little bit later about the Gulper sharks and why that sample size is so small. So we estimated that post-release mortality rate. So to address that first objective, What we did was calculate it using the cage as the replicate. So you might think the easiest thing to do would be to take the total number that died out of the total number that we sampled, but that doesn't allow you to calculate any kind of error. And so instead, we actually took the sum of mortality rates across all cages and then divided by the total number of cages. So we get that standard error. So for Gulper sharks, we found an at-vessel mortality rate, meaning the number of individuals or percent of individuals that were dead when we first got them at the boat. And for these Gulper sharks, it was pretty high, it was 30%, really similar to a study by Brooks et al. At the same location in 2015. And for the 24-hour post-release mortality rate, it was 83%. All of them died except for one. They all exhibited the same response in the cage after our release. And the one that did survive actually was in terrible shape, but we could see the spiracles still pumping, so it was still breathing, but it fell out of the cage door as it was hauled to the surface. So if I had to take a guess as to whether it lived or died, I'd say it probably died as well, although we didn't document that. For the Cuban dogfish, at-vessel mortality, about 8%, a bit lower than the gulpers, very similar to studies by Brooks in the same location, and then Gulak in the Gulf of Mexico. The 24-hour post-release mortality rate for that species was about 50%. If you take anything away from this presentation, that's probably the most important finding that we had over this work over the few years. Basically, this can be directly implemented into that equation for total fishery mortality for animals caught on long lines and then subsequently released. And it's kind of interesting that it's 50%. It's about halfway between the assumption for long line and trawl fisheries. So because we have these video cameras in the cage, we also look at cumulative mortality over time. You could actually see when animals were dead. And so this is our death plot here. So we've got time post-caging, so basically zeros when the cage hits the floor. And then this is 800 minutes later. And so you can see basically for these two species here, that by about 700 to 800 minutes, like 11 to 13 hours, all of the mortality that we saw within 24 hours had already taken place. So mortality takes place pretty rapidly after release. That gives us a bit of comfort in thinking that we didn't actually underestimate mortality by only looking for 24 hours. If they were still dying at 23 or 24 hours, maybe we should have monitored them longer. But we think we captured the bulk of it with our methods here. So we also wanted to develop a method to predict mortality so that future studies can actually just take a blood sample or measure a few things on the boat and then actually predict it without monitoring the animal after release. So we did that for the Cuban dogfish, again, because we had a good sample size there. I'll give you a little background on stress. The stress response in elasmobranchs is kind of similar to in us. Basically, there's this capture event or the stressor. The animal has this fight or flight escape response, much as you or I would if we got startled. And then the kind of suite of changes in the blood and behavioral changes can be broken up into these three categories. Primary response refers to a release of hormones into the bloodstream. So just like us, it would be adrenaline, noradrenaline from kidney tissue. And then that translates into the secondary response, which is basically all the stuff that happens as a result of those hormones entering the blood. So for instance, release of glucose from the liver to fuel active muscle tissue, things like change in heart rate and ventilation rate, and the change in blood gas concentrations as a result. That's a secondary response. This is actually what we measure in studies looking at stress in sharks, because the primary response, for a teleos, you might want to measure cortisol, which is great. For sharks, their primary stress hormone is something we don't have an assay to actually measure. So instead, we measure these changes that take place afterwards. And then the tertiary response refers to the organismal level response. Does the animal live or die? Does it experience an impairment in immune function or reproduction or growth? And so we were looking at this whole picture here. So the way we did it was we took blood and then we looked at various factors as well as total length to see if we could actually predict an individual's fate based on those metrics while still on the boat. We used the binomial distribution for GLM, threw all these factors in. Binomial response was what did the animal live or die after 24 hours. And we found that blood lactate, glucose, and total length were the significant predictors. And we created a model using those three variables to predict mortality. I'm going to talk about just lactate and total length because they had the largest effects. So we have models for each of these depending on what variables are actually collected in a fishery setting. But this graph here, you have probability of mortality on the y-axis, 0 to 100. And you have total length going from large to small, and then lactate from low to high. And you can see that the effect of total length is such that the smaller the animal, the more likely it is to die. But that effect kind of pales in comparison to the effect of blood lactate. The higher the blood lactate, the more likely the animal is to die. So I'll kind of explain that with some visuals here. This is a Caribbean reef shark that's just taking the hook, and you can see its response to capture is very pronounced. There's a lot of burst swimming. These animals are made up of a lot of white muscle, and so changes in the blood, changes in behavior and in the tissues are reflected in the bloodstream, and they're really not meant for sustained burst swimming. So it gets stressed very quickly. And as you can imagine, just like us, if we go for a run, carbon dioxide is going to go up very quickly, oxygen is going to go down, lactic acid is going to build up. All those same changes are happening in the shark here and in our deep water species. Basically what's happening is they're shifting from aerobic to anaerobic metabolism as they don't have enough oxygen anymore to create ATP. And this is reflected in the blood pH. So here we have pH. Basically, the lower it is, the more acidic. Then our two groups, those that survived and those that died. And this is for our 24-hour cage result. So lactic acid and carbon dioxide basically are being elevated in the bloodstream. That results in blood acidification, and that's evidenced here. Basically, animals are more likely to die if their blood is more acidic when they're at the boat. So all of these things kind of come together to present a complete picture. So we can predict what happens to an animal based on the secondary response. Can we do it just by kind of eyeballing the animal? Maybe it's too difficult, too much to ask, but in a fisheries context, we would collect a blood sample. Can we do it another way? So we came up with these vitality scores from excellent to poor. Basically describes body movement and injury. Basically poor, weak, or little body movement, and maybe some injuries, maybe some bleeding from the hooking location. We found that we could actually predict it just through those kind of visual methods. So here's 24-hour cage result, 0 to 100. Basically, for those assigned a score of excellent, 20% died, 80% survived. Assigned a score of fair is about half and half. If they were assigned a score of poor, they all died. And so that's really interesting because even without taking these blood samples, we can potentially predict mortality just with this kind of rough metric here. So I'm kind of running out of time, but I want to talk briefly about post-release behavior. We had these three objectives. We first wanted to link blood chemistry with post-release behavior to get at the sublethal effects as well as the lethal effects. Again, I want to talk about these gulper sharks so I don't conveniently ignore them completely. But we found this 83% post-release mortality rate, and that's using this method. So we think that the cage has some kind of impact on how these animals are responding because they all hover upside down. They're unable to orient within the cage. They stop swimming. They still survive for a while, but they do end up dying over time. And we're really not entirely sure why that happens. We think it's related to both the effects of the cage itself, as well as potentially really high capture temperatures where we were doing our study, because some of them in a study in Australia actually survived, and those were caught at much lower temperatures. So there's a lot more work to be done for that species. So we're just going to talk about some of these for Cuban dogfish. Here again, we've talked about blood lactate going from low to high. Basically, as you go up, the more stressed the animal. You can see for animals that were inactive, meaning they spent less than 20% of the time in the cage swimming, they had higher blood lactate levels at the boat. So what does this mean? Well, for me, it means that basically the more stressed the animal, the less it swims after release. What are the implications for that? Potentially, they're more susceptible to post-release predation because their swimming behavior is impaired. So maybe post-release mortality is actually higher than what we're estimating. We also looked at the effect of depth and temperature on recovery. So here, just to remind you, as depth increases, the temperature decreases. It gets much colder at a predictable rate. And so we looked at the recovery rate of sharks that survived between shallow cages in the light gray and deep cages in the black. And so we were looking at this as mean time swimming over these video segments. You can see that the rate of recovery was higher for sharks that were in shallower cages. Why would that be? Basically, we think it's related to metabolic rate. Sharks, their metabolic rate is tied to the temperature of the external environment. And a convenient study actually looked in the California bat ray. A 6-degree temperature increase resulted in a 3 times increase in metabolic rate. And that's convenient for us because the difference between our deepest and shallowest cages had a 6-degree Celsius temperature difference. And so it makes sense. Basically, the sharks that are a bit deeper in colder water take a longer time to recover from the stress of the capture event because they're basically slowed down. It takes longer to get back to physiological homeostasis. Last one here, the implications of post-release behavior. We did monitor some of these sharks that we released at the boat using video cameras and some other things. And you can see their behavior is not what you expect. They don't swim down to the bottom. Again, they have a long way to go. Any kind of counter shading that might help them kind of blend in and not get seen by predators is ineffective, as they're basically rolling all the way down. Again, they have some physical injury, and there's no cover. That's a lot of wide open blue water that makes them very susceptible to predation. We tracked 4 animals. We tried to have like a side study. It didn't work out that well. But for 4 animals, one of the 4 got eaten by this silky shark. So here's the rate of descent for, the average rate of descent for our cages. It goes down quite fast. Here we tracked this shark from 0 to about 130 meters, and it took 15 minutes for the animal to get there. Really long time. So it's susceptible, its profile is very visible from below during that whole period. And indeed, a silky shark did eat it, and we caught that silky shark somehow. It's kind of a weird story. But so impaired swimming behaviors on the way down, very risky. Then they get to the bottom, and they encounter another suite of predators. Predators that you might not think of when typically we think of sharks as apex predators. That's not always the case. So here are 2 sharks. These are Cuban dogfish after about 22 hours. They're in really good condition. They've been swimming around happily. The door falls open, and in comes a giant isopod. Imagine a roly-poly from your backyard from hell. So this giant isopod has 4 sets of jaws, very efficient at ripping flesh off of animals. And you can see, we were watching this, and it kind of hopped on that shark. And then it finds this other one that's just chilling in the corner. So that happened. The isopod ended up killing the shark and taking its body out of the cage. That was the first time this was documented. We thought it was pretty cool. Point being, I don't know how often this happens, but I just want to show you the video mostly. Also, they not only have to escape predators on the way down, but when they get to the bottom, the story's not over. And if their behavior is impaired, if they're not swimming as well as normal, they're going to be resting on the bottom, and that makes them susceptible even to these roly-polies. So there are limitations to our study. We think that we're probably underestimating post-release mortality because we're excluding predators from gaining access to these animals. You could make the case that the cage is actually doing the opposite, but we found that there were very few cage effects. Animals didn't have any physical injury or anything from the cage. We don't think it made them worse. And then there's this issue with the gulper sharks, so we take that rate with caution. So to wrap it up, we can predict post-release mortality and post-release behavior for the species of small dogfish that is caught as bycatch in the Gulf of Mexico using these 2 methods. Capture and recovery depth can impact behavior and survival. It might be slower at greater depths. We think that we're underestimating these post-release mortality rates. We think that it could be quite common. And most importantly, deep-sea sharks are likely experiencing far higher levels of post-release mortality than currently assumed in many fisheries. Again, for the dogfish, around 50%, for the gulpers, a bit higher. So with that, I want to thank, there are so many people that worked on this project, down to Cape Luther Institute, Island School, all the funding sources, and of course my advisor, Dean Grubbs, as well. So with that, I don't know if I have time for questions, but